So, good evening everyone. This is Shubhadeep Ghosh. I am MSc postgraduate and worked as Junior Research Fellow in Zoological Survey of India and worked as contractual basis guest lecturer in Rohra Ramakrishna Mission, Vivekananda Centenary College. Today I am going to talk about osmosis, one of the most fundamental physiochemical process in the plant and animal cells, which helps in nutrient balance, water balance and excretion in the animal and plant cells. Okay. So before we are going to talk the main topic, let's see a basic concept, which is a solution. So we can exemplify this by using a sugar solution, where we put a teaspoonful of sugar into the glass of water. So what will happen? The solute, which is represented here as the sugar, will get dissolved into the major part in the glass of water. So the water will be the solvent and the solute will be the sugar. So this is the basic part or the basic theory or the basic concept of on which our main topic of discussion, osmosis, lies. We are going to talk about osmosis in the next part. So what is osmosis? Osmosis is the movement of only solvent across the semi-permeable membrane from high to low concentration. So this picture represents the solution, where the black dotted one represents semi-permeable membrane, and the blue circles represent the solvent molecule, which is getting passed through due to its smaller size through the semi-permeable membrane from high concentration to low concentration. But due to its larger size, the red molecule, which represents the solute molecule, cannot pass through. Okay, so this is the main basic process or fundamental process of osmosis, and according to which we can divide osmosis into two parts, which we are going to talk about in next part or next slide. So in the next slide, there will be two types of osmosis. Okay, two types of osmosis represented here in the third one and the first one. Okay, if we put a kishmish into the water solution, what will happen? It gets swells up. Okay, so there will be a structural change in the kishmish. So in this part, the water is coming inside the kishmish, in the dried kishmish. Okay, in the water, when you put into the water, so the coming inside of a solvent into the kishmish is called as the endoosmosis. Clear? And the swelling up to a certain level of the particular kishmish can lead to bursting out. And according to the kishmish, there will be higher amount of solvent outside the environment. So the solution for the kishmish will be the hypotonic solution. Okay, and in the first part, if we put the swelled up kishmish before bursting out in the sugar solution, what will happen? There will be very less amount of water outside the kishmish, because there is a sugar solution. We put a sugar solution, so the water tends to comes out from the swelled up kishmish. Okay, so and the kishmish gets shrinked. The structure will get the structure of kishmish gets changed and it gets shrinked. So according to the kishmish, the solution, the sugar solution particularly, is the hypertonic solution. So this coming out of water is termed as exoosmosis, the second type of osmosis. So always remember, exo means something is coming out and endo means something is coming in. And now coming to third part, where the yellow represent the coming in inside the cell and green represent coming outside. So the amount of water coming inside equals to coming out, equal amount of water coming outside. There will be no structural change in the kishmish. So where the solution, where the exosmosis rate equals to endoosmosis rate is equal, the particular solution is termed as isotonic solution. The particular picture represented here by using the RBC cell, which is quite similar with the scenario of putting kishmish or into the sugar or water, both cases. So how it is different from diffusion? In particular osmosis, there is a physical barrier, which is represented here as a semi-permeable membrane, which possesses a particular size pores through which only solvent can pass through. So there is no chance of solutes can pass through the semi-permeable membrane. But in diffusion, there is no semi-permeable membrane. There is no physical barrier. So the molecules of both solute and solvent can pass or intermingle with each other, or can move easily from high to low concentration. Okay. So what are the significance of osmosis? Osmosis helps in maintaining water and nutrient balance in cell. It also helps in excretion in the cell by the particular part which I have talked about, the movement, the specific type of movement. The smaller excretory molecules can pass outside the cell. So this, by this osmosis process of osmosis, a cell also takes part in the excretion process also. Okay, so it helps the, by the process of osmosis, it helps in maintain the turgor pressure, which is the shape holding nature of the cell. So it helps in receiving water from soil via roots in plants. It is the main process of kidney dialysis. It helps in germination of seeds in plant. So these are the most fundamental 5 functions or significance of osmosis, and we can easily say the osmosis is the most useful process for both an environment and plant cell. Okay, so thank you.